Well, or we just get going with the, well, it's just, which we're fine. I think then we have more time. Hello, everybody. It's a pleasure to see you today again. And now we will get started with the report from the breakout sessions that were very lively. The first, I should just say, I'm Genevieve Almouzi from the Curie Institute in Paris, and I'm delighted to be able to hear and learn so much from all of you. So we'll continue on that note with Anne and Sten, who will report on the first breakout session. Please, the floor is yours.
Okay, so we would like to first thank all the people who were at the session, because without a lively participation, you get nowhere in the discussion. And our mission was to discuss regulatory mechanisms in development, understanding seller decision-making during development. So to get the discussion started, we started with a slide which put very traditional questions in developmental biology related to decision-making, asking whether these questions were sufficient, whether the audience had others, whether they were still relevant in the context of human development. We started with the idea that development starts from progenitors, and they have a certain, they have a certain potency. And I'll come back to this question of potency. That then they differentiate and they make choices with branching points that can be binary, or we may consider as more than binary. This is submitted to extrinsic and intrinsic controls, extrinsic coming from the cells around, and then the cells differentiate. This differentiation may lead to a point of no return. We discussed whether these are steps or a continuum, what is the speed at each step, and whether these decisions are stochastic or deterministic. Now the big question, since we are a cell atlas with a lot of molecular data, is how to link gene expression and cell behavior. We also discussed maturity, and initially on my slide I had put maturity here singular, but actually if you consider the cells that mature, it's rarely singular, because even you have to go to the adult, and the cells may be in different states. So we don't even know what the end goal is in this diagram. Now, what we also discussed is that all these decisions need to be put in the morphogenetic space. The cells are not alone. They have neighbors, they have matrix around. So the first question we addressed was potency. And we realized that our definitions were quite variable, and that it's linked to plasticity. One possible definition is potency is what the cells can become, but they don't always become that. It depends on the environment they will meet, whether it's the in vivo environment or an in vitro environment that may reveal something they can do, but they don't normally, as we heard, for instance, in Prisca's talk. Traditional assays have been clonal growth and differentiation assays, as well as lineage tracing. But what we realized with the Human Cell Atlas is that we should go to quantifying. So how would you quantify the potency of a cell? One suggestion was that it may be the ensemble of signals a cell can respond to. We may want to also monitor the strength of responses. This should allow us to predict from single cell data, taken into account the cell, the progenitor at time t, but also its history. And the history from the data at the time t is not very easy to infer. And the environment, which is probably easier to infer that we can do something with, meaning other cells in the matrix. then the discussion moved to a question raised by the audience, is now that we have a lot of data, can we just predict cell decision from single cell data at additional models? So there's certainly a need for models, but there are also current limitations that made us doubt that just with models, we can understand cell decision making and predict everything. First, because single cell data mostly inform on the state of a cell at time t, but it's less easy to infer the history. And one of the things we discussed would be, well, would the methylome at the single cell level be a step towards informing us about the history of the cell? We realized that ATAC-seq is not sufficient because even though promoters may be open, some may not be open and can become open when you are in specific conditions. We also discussed that beyond transcriptome, we certainly need protein data. And one of the approaches discussed is mass spec on section. The resolution may not be sufficient, and that AlphaFold to infer protein interactions in network may help us. Now, a major block to just work on inference of cell decision making from data is our difficulty to deal with combinatorial analysis in computational modeling. And that's something that further developments would be worth doing. Maybe I hand to Sten.
Yeah. Right. Yes, so we had the discussion also on what kind of new tools we need. Certainly we need more comprehensive data, but there are certain challenges, in particular in human tissue, that make it very difficult to do true functional experiments. But approaches to that might include, for example, explant culture. It's possible to grow human fetal tissue for several days, maybe up to a week, which gives a possibility for perturbations. You can also use model organisms. There are exciting, I don't know where I should stand to avoid the echo. At the podium. Okay. No? No, okay. Explant is one thing, but there are now exciting new methods for ex vivo embryonic development, so ex utero embryogenesis. And another exciting possibility is to use transplantation chimeras, so either transplanting individual cells or transplanting, for example, organoids into model organism tissues. Maybe we can take the next. Oh, that was all, okay. Okay. I'll go from the notes then. I think there was one very important and I think exciting suggestion from the audience to make use of cases where human development goes wrong. So abnormal development as sort of natural experiments, and if you would combine that with genotyping, you might be able to relate changes, for example, chromosomal aberrations or natural knockouts to developmental abnormalities. But this poses very challenging demands on sample collection and being able to systematically collect such embryos. But I think that's an exciting and important avenue. All right, and then of course when we use all these model systems, various approaches outside of using pure human tissues, there's always the question of fidelity and how much our models recapitulate human development, normal human development. And so that's an important role for the Human Cell Atlas to provide sort of ground truth of normal human brain development or human embryonic development, so that we can understand whether our organoids and our transplantation experiments and so on truly recapitulate human development as far as it goes. For example, when you transplant human neurons into mouse brain, the human neurons continue to develop on a human time scale. So they go very slowly in the mouse brain. And so therefore it's difficult to model. In a sense, you get a mismatch between the systems. So maybe there is a need for developing matched model systems that develop on a similar time scale to humans, rather than using the rodent models, for example. What else? Yeah, we discussed the importance of obtaining clinical information on donors when you work with human samples. This is always often difficult to get, but it might, of course, affect development. The context in the womb of the developing embryo is of course, incredibly important for normal development. And it will also be important to look at things like sex of the embryo, for example, and keep track of that. Another in discussion, maybe going back to the first question, is about our ability to obtain quantitative measurements. So if our goal, for example, on the question of potency, if our goal is to predict what a cell will become or could become, we'll need certain kinds of information. And we have very good measurements of the current state of the cell in the form of transcriptional state and chromatin state. But we have very feeble measurements of the current environment of the cell, which of course is super important. So we can, to some extent, infer what the neighbors are of a cell, but more specifically, in a quantitative manner, understanding the context of the cell in terms of signaling proteins, morphogens, extracellular matrix, and so on. There, I think we lack, we really lack tools, and what we need are quantitative tools that can tell us concentrations of proteins in the tissue, because the ability of a morphogen to activate its receptor, of course, depends on the local concentration. So quantitative tissue proteomics.
Okay, well, thank you so much for this great summary. I was there, so I can attest. It is really a great summary. So we will now move to the next reporting, and I think it's Stephane, Nedelec, and Prisca. Try to pay attention to really speak close to the microphone, because for the hearing, it's not working great otherwise. So I can hand you mine if...
Good morning, everybody. So yesterday we had our breakout session with the organoids and animal models. Oh, now it's working. And I think the general framework we use to start is to try to understand which in vitro model are generally used and how we can use them, what they're good for. And I think that was already to get, on average, what people in the room are using. I think there are very different models. So I think there are the models from iPS-induced cells. So from iPS cells, we can make organoid from very different tissues. The problem of this organoid, they are like still in a very fetal state. They recapitulate a lot of development. And so this is one type of organoid, the one from iPS cells. And then there are the organoid from adult stem cells. For example, intestinal organoid are used very much, and these are cells directly from mouse or in mouse, or from human and directly from patient to create organoids. So it was clear that most of the people use the one from iPS derived, and this recapitulate a lot part of the development. And I think they were also trying to differentiate different steps. One part is really the embryonic to fetal, but also the fetal to adult. That right now is still not really accessible, because with organoids, especially the iPS-induced one, we are in a stage where they stay very fetal-like. There's really many people that are currently trying to move the fetal to adult, to understand what is bringing these fetal cell types to adult cell types. And I think these will be really important for the future. So they also, how accessible they are for imaging, for perturbation, and for both multimodal and multi-scale analysis. And they are tractable. And clearly, they are enabling us to reach some aspect of development that are not accessible because we cannot get on one side the samples, or also for disease state. We had a very big discussion that some diseases are really not accessible. And with these organoids, we can go and see when is the onset of disease. And so the perturbation, and I think the part of the translational research, and this I think we'll go back also at the end, is like, when is it a good time for the Human Cell Atlas to start looking at that? And then I think, as similar was also mentioned before, on abnormal development or disease state, when is the time to understand these? And then I think we'll go also back in the next slide with it, but I think this was a very big part of our discussion. So the main point we tried to discuss was, what is it needed to move forward? Where are we now, and how can we move forward as a community? How we want to solve some of the problem as a community, and then how the Human Cell Atlas and the Human Cell Developmental Atlas can really be helpful for that. And so I think here we'll have 3-4 points that we have been discussing. The first is the multidimensional studies. And I think it's very important because we start often from these iPS cells. Which cell lines are we using? And this is information that is very different, very important to share, because different cell lines have very different potential to make different organoids. They can be, some make endoderm very nicely, others make mesoderm, others make gut. So I think this will be very important to look at cell types that are genetically stable, but also like for modification and for screen. And so I think we start doing a lot of measurement, which cells are we using, and which one will be used also for the Human Cell Atlas to have this information. The other aspect that we discuss is the protocols. Right now you can probably go online and see millions of protocols for any of the organoids, and so which one is the good one? And that will go to the second point, that is really the benchmarking, how we can define a good protocol, and especially when we want to increase the complexity of the models and have different type of organoid together or co-cultures, what are the standards? We cannot start having 5 different protocols for an organoid and then add the complexity of 5 different protocols for the co-culture. So what will also the Human Cell Atlas or other part will be for benchmarking and to have this? And so I think this is a very big problem, also because not only the cells, not only the protocols, but the media is very important in this case. And for example, there are a lot of companies that sell medium, and we have no idea of the composition of these mediums. And so if we then start using those for the benchmarking of our organoid and the medium change, we're already changing everything. So I think maybe even as a community reaching out to some of the companies that at least some aspects of their medium would need to be shared and open source for integrating, especially if these become the core of our let's say, benchmarks and then our organoid lines. The other one is the main issue we have been discussing. Some of these protocols are very long, and it would be very important step that could be improved. For example, if we have a protocol of 5 months, could we have freezing step in between, that at least you freeze after 2 months and then you get a very good bank of frozen samples, so you move and you have only 3 more months to go for growing them. So I think this is where a lot of people are working to improve the protocols, get them maybe faster. On the faster, I think we don't all agree, because probably some parts need the self-organization. And we had that discussion on timing. Is this similar to development, or can we really accelerate human development in vitro in a way? And then how much is then recapitulated? But I think these were part. I'll start maybe the benchmarking, or you want to take over? No, I don't know. It's like, because then there was the benchmarking part.
Okay. So yeah, I think one of the key points that came out during the discussion is how, so if we can decide on specific protocols, specific lines, how to benchmark the models that are used, and critical will be to define divergences in the in between the models, in vitro models, and the in vivo cell type from embryo or fetal cells. And comparison to human embryo will be obviously critical, and it was mentioned in the previous talk, to define similarity and divergences in terms of cell state. So even if a cell is identified as a specific cell type, is it how much it compares to the in vivo cell types? And also within specific organoids or more complex model, how the diversity of cell type is represented, the missing cell types, the ones that are overrepresented, as well as their spatial organization. And that it came out as a very important that HCA could have a critical input on that by sharing, of course, the data set that we'll be generating to compare the organoids, as well that the pipelines of data management and analysis that will be very important. And as mentioned, the need for late fetal stage in comparison to adult will be very critical to push the maturation of this organoid. And there was a proposal, interesting, it was to integrate the organoids in the HCA biological network to properly annotate the cell types within these cells. And the idea was also maybe to, if the biological network of the HCA are missing developmental biologists, to really bring people there to help annotate the early stages and the developmental trajectories that could be very essential to benchmark the organoid model. And finally, if I have 1 or 2 minutes, we discussed the question of the complexity of the models. So organoid models model single organ or a fraction of it most of the time, while development, disease, and drug actions act at higher scales as well across organ. And so there will be a need, and several people brought that during the discussion, of the complexification of these models. The opportunity to define condition, contribution of other cells, the type of organ, of other organ on cell behavior, cell identity and diversity, as well on morphogenesis. The importance of the niche also in this diversity, and how immune cells could be important for this aspect. So it was discussed the issue of how to build this more complex model, how to standardize them, and when to integrate them in the HCA. And the different approach that have been discussed is the embryoids or stembryoes that are so-called the assembloids or organ on chips, and when can we bring these models and what they can provide and how to really validate them. And finally, last question was when to include disease state and with which models in the HCA initiative.
Well, thank you. Perfect, on time. So we continue with the reporting before the questions. So I think it's now Christel and Stephanie, if I'm correct. So please. The floor is yours, and try to speak close to the microphone.
Okay. Good morning, everyone. Can you hear me well? Okay. So we had this breakout session on developmental disorders with Christel. Unfortunately, our colleague Guillaume Canaud couldn't come yesterday. And so because Christel and I have both a background working with genetic brain disorders, I have to say that the session was a bit oriented toward brain disease. So can I have the next one? Is it me? Okay. So, and also I would like to thank the participants for their helpful suggestions and the nice discussion that we had yesterday. So we pulled up the main aspects that were discussed yesterday, so I think we will be quite brief. Okay, so basically, So basically, the main question here was how can we use a comprehensive reference HCA maps to understand disease mechanisms, predict disease outcomes, and develop some prediction models. And so within this big question, we had several subtopics. So one first aspect that we had in this session was talking about genetic brain disorders, either due to constitutive or to somatic mutations. And in somatic disorders, as you know, only a fraction of the cells carry the mutations. And so there's a challenge here to identify which are the cells carrying the mutation and that might be abnormal. And so can we identify those mutated cells within the HCA maps in the context of mosaic disorders? And with this, the question was, are they, do they have like a different cell type or rather a different cell state? And that's kind of one of the challenge. And the second challenge here in these mosaic disorders is to identify the mutations within these data sets. So how can we combine 10x single nuke RNA-seq with sequencing toward the whole transcript? And this is something that emerged, that we need new tools to combine both sequencing aspects. The second big question was also, how do we define and set a limit between what we would call normal and abnormal status? And of course, there's a whole continuum, and the challenge is to set up this limit. There's a huge variability between individuals, but also toward a single genetic event or environmental event. And the challenge is then to take into account this variability, where we know that between the transition between asymptomatic to symptomatic, where can we put the cursor and identify this change? So yeah, with the transition between asymptomatic and symptomatic status, there's a huge question. So we had a discussion on neurodegenerative disorders and with the case of Huntington's disease. As you may know, it is now known that in the fetus, so very early in the prenatal development, there are already some hallmarks and markers of the disease, while the symptoms will appear only 40 or 50 years later. And so how can we use those HTA maps to identify before the development of the disease some markers? But in the meantime, for neurodevelopmental disorders, it's the contrary. We know that we need and we lack some tissues, especially the third trimester in pregnancy, for instance. And so how can we fill those gaps? And of course, there's the need for iPSCs and organoids to have a continuum in the different developmental stages. And with this, we need to predict the key developmental windows for the disease, to catch up also all the rare or the transient cell types that are present only in developmental stages and that are not present in the adult. And with this, to design some targeted therapeutics with cell type specificity. So can we, once we identify some abnormal cells, can we target specifically this cell type? And when, of course, do we need to target them if we know that the disease will occur along different years? So Christel will get the next channel.
Okay, good morning. Yeah, so you heard from Stephanie, so we discussed the need to fill in gaps for missing data. That's for obviously, so we'll have data missing from, for example, fetus from the third trimester. So we discussed the possibility to use organoids or animal models to fill in the gap. And one obvious point is that we have to choose the appropriate model to do that. So for example, if we want to model anomalies of the corpus callosum, which is a commissure only present in placental mammals, we can't use species like xenopus or zebrafish because they don't have this structure, so we have to use a mammal. On the other hand, we can use xenopus, or we can use to model other disorders like eye development. So xenopus seems a very good model to model eye development, for example, because it's a more evolutionary concept. We discussed also that we really need to have a common language to describe cell annotation, to describe like very precisely markers defining a cell type. And there is really some work to do to achieve that goal. One limitation is that one cell type in one species and one cell type in another species might be different, obviously. So there might be more cell types in humans compared to some models that we use. So one way is maybe to use evolutionary conserved networks to address this question. And that would be very, very good if we could get a tool like BLAST some kind of tool where we just do a query to say, okay, that's my markers, they are specific for my disease, and then we want to know the cell types that are affected in this disease. Then we also discussed the fact that we use the human reference genome that is quite old, which is mainly AG38. So as you all know, there are a new reference genome, or now more than one actually, that is complete, with more genes, like hundreds of new genes, and also repeats that are annotated. So we probably need to move to that reference to get a better overview of the cell type. Then we also discussed the transcripts. We know that there are human-specific transcripts that can be transient during development, that can be very important. And to really look at those, we would need to use some long-read technologies to really have all the human transcripts that might be important for human development. And then again, I mean, Stephanie already touched upon this need. We need to understand diseases, we obviously need to understand what is normal, and that's a very difficult question, because we know that there is a huge variability between individuals, and the question is how many maps actually we need, probably not just one, to describe what is normal. So this is a question, an open question, but we probably need more than one. Yeah, so we would like to have a feedback on you know, please ask questions or about this session, and also come and discuss with us at the breaks. Thank you.
Okay, so I guess what we should do now is open up the floor for the discussion on all aspects related to the breakout. And it would be, of course, very nice if the people that have been leaders of this session can be around to help also contribute to the discussion that we can have now. So please, if you can join me, and yeah, whoever is happy to get going, and even between you, because you are leaders of sessions, but you could interrogate or have questions for other people within your own session and out. Okay. Yes. So please go ahead.
Hi. So regarding the new reference genome, I was just checking now also, even in Ensemble and in CBI, but they are still not available there. So how would you recommend to start using that infrastructure if we still don't have the tools? Especially because Ensemble, most of the tools are in Ensemble and CBI. And of course, all tools are already connected to those databases. If the genome is not there, how would you recommend on that?
Yeah, I completely agree with you. So it's a transition period. So obviously this will be available. I'm not sure when. So now that it's available on UCSC, we can use it. We can lift over the coordinates. I think that might be the best way to do it now. Like map on AG38, map on T2T, and use the lift over to annotate. But I agree that's not that's not perfect.
So this is actually raising an important issue for a transitional period. If there are any recommendation within HCA to consider to harmonize the way to do things, it does ring an important bell that we should probably think about. Indeed. Now, other questions, comments? With respect, yes.
Thanks, Genevieve. Thank you very much. Perhaps it'll be interesting to have an idea of the logistics of integration, because there's a lot to do, and there have to be priorities, and we talked about benchmarking, so that's clear that we need in vivo signatures, not only of the human, but the animal models, because we're coming up from animal models. But then if we want to project onto that the different aspects that all of you covered this morning, the organoids, etc. How is that decided? We know that there are variations in protocols from different labs, and people will fight tooth and nail to say that theirs is the best. So how is the triage going to be done logistically?
Okay, so that's a tough question.
I don't know.
Well, I think it's a very reasonable answer.
I think at this point, it's still there have been some exploring some of the aspects on because now the organoid is still like a separate network. So there's the HCA organoids and how to integrate the organoid in the biological network, and if to integrate it in the developmental one. So I think there is still quite some work, especially on the organoid part, to see where is the best way to integrate it. And I think to have it them alone, it's really a bit of a problem, because then we might be increasing then we'll need a biological organoid network. Do we really need a biological organoid network, or can we integrate it in the biological networks? And there's a part on organoid there. I think that might be the best, because at least there are some experts, as we said, some networks for the integration.
Yes. Yeah.
But clearly part of the organoids go back to the developmental one. So do we want to have, where do the organoid fit? Because part of it, so for example, so yesterday we didn't discuss anything about cancer or canceroid, this type of disease, but
yes,
clearly there's a very big field there too.
Totally. And I think that has been also brought up. I think
we have a better Sarah.
But I think it is a very important point that we need to really work on. So Sarah,
okay,
please.
No, so to the point about sort of integration and I mean, what I'll say is that we are only now at the point where we are sort of assembling the human cell atlas, only even at the level of suspension cell and nuclear data, I would say, not even properly spatial data. And where that effort is kind of most advanced is in the adult organs rather than development or organoids. And in the adult organs, basically, there's now 12 postdoctoral positions funded by CZI, where each one is basically focusing on one organ and integrating all the published data and any unpublished data that's given, for the gut, for the brain, for the kidney, et cetera, et cetera, et cetera. And then, the next logical step would be to go to, extend that into development potentially, and then sort of do the comparison with organoids. So I'd say one prerequisite, if you like, is basically the having a gold standard integration and having a gold standard kind of consensus cell annotation as well, and that's part of all of these efforts. And then the next step, is basically ways of quantifying similarities, basically, and there are a bunch of different computational approaches for that, including, logistic regression, weighted nearest neighbor, label transfer, et cetera, et cetera. So I think there are multiple components in that question, and there's kind of a stepwise sort of evolution towards that, certainly. And I think it's fantastic, to think about, as Prisca said, like, is it that each organoid community should be linked to that organ in development and maybe all the way through to adult? Maybe there should be a connectivity that's kind of orthogonal, to organoids. Because at the moment, it's, we have communities organoids, we have development, which is us here. We have then the biological networks in the adult. And definitely, there can be like orthogonal connectivities across the biological tissues, organs, et cetera, for sure.
Can I just follow up a question?
Yes.
And maybe Sarah can keep the microphone because it's to Sarah. So, because then clearly being a community effort, and at least in our discussion, was like, how do we decide which one is the good protocol to start having some data? Because clearly we are far for being able to integrate them all, but we'll start generating all the data. So how to make sure that some generation of data is done in a standardized way, and especially I think with organoid, in a way with human tissue, it's already getting some human tissue is already incredible. So while with organoid, there will be an explosion. So how can we make, as a community, even as just the organoid, making some benchmark already now, that then when there will be the effort in 5 years of integrating, we are not realizing that we have a lot of crap.
So I think, you know, I obviously don't have like the perfect answer for that, but from a practical point of view, the way I see things going in the community is that the human cell atlas data, you know, is being used to benchmark or I wasn't in your in your discussion, I was in the regulatory networks discussion, so I don't know what you covered, but the way I see things going is that the cell atlas development and adult data and so on is used to benchmark and compare different protocols. And then the way I think about it is that the human cell atlas provides the recipe, if you like, for guiding, you know, the programming of the protocols. I mean, that's obviously I'm looking at it from a human cell atlas perspective, but and maybe there needs to be an iterative process, you know, that you have the current protocols are benchmarked, and there's then like a decision of, okay, we're going to modify in this way, kind of guided by the in vivo data. I mean, that's one way of thinking about it, but I'd be curious to hear others.
This is also part of the way we discussed it yesterday on trying to use those data as a benchmark and have these similarity scores. So having something saying, okay, the problem is that, for example, there might be very good protocol for endpoint, that then they recapitulate really well the endpoint, but maybe for development, there might be a protocol that recapitulates better development. So I think there will be part that where biological network and endpoint for a certain state will be better, but clearly the human cell atlas, I think, would be great for benchmark.
Yeah, well, that fits really with the kind of organoid that you've been working on, where the endpoint is fine, but to access to some of the intermediate things, you still don't have the proper model. So, Anne.
Yeah, I like Sarah's approach of trying to map the protocol to a space where you see the in vivo development, because I think choosing a protocol that will work for everyone is not an easy thing. First, it depends on the line you start with, but also if we go to disease models, what we see is that there are protocols that tend to mask the effect of mutations and protocols that sensitize where you see the effect of mutation much easier. And I think it's important to keep the flexibility to explore this space.
So I guess your point, Anne, is really important, and we need to reflect on how best to sort of go back and forth with what you want to model. And if you want to model a particular disease, then there may be some requirement that could be distinct. So we probably need to have things where the dimension of the disease is now perhaps better integrated along with the development and the endpoint tissue as well. So perhaps for human cell atlas, it would be good to think about that, because we've been talking about integration as a major aspect for the data, but it's also a major aspect in the way the questions are developing at this point in time, and it would be probably a missed opportunity if this communication is made too late, and so people do things a bit at random. So at least knowing that there's a way to talk to each other and exchange on this aspect would really help. Further questions, comments, ideas? Yes, Gary.
I just wonder, from the diverse topics that we covered at the breakouts, I know there was some mention of cell annotation. So right now, people are focusing mostly on transcriptome profiles. So what would be the ideal types of information that we would need to best identify cell state, cell type, not just at the transcriptome, so functional, spatial, what kind of information do we need to think about combining to make that definition?
Another big question.
No, one answer in brain cells is, of course, to do patch-seq combined and spatial transcriptomics, of course, because we know that the position, will inform also on the function
Yeah, I also wanted to say that for development, of course, spatial information is even more important than in adult, at least for the brain, which is what I know. In the adult brain, the position of cells doesn't really matter very much. It's all about how they're connected to each other. But in development, the position is everything. And so I think we need to try to go from 2D spatial to proper 3D spatial so that we have intact coordinate systems that are sort of the natural coordinate systems of the embryo. And then we need to measure what I mentioned before, quantitatively measuring the environment of the cell, especially morphogens, so we can understand who is talking to who and who is receiving the signal in the 3D context.
So that's an important point, because the development of the technology on that front is really up and rising.
For development, do you think we need to also consider the vector of potential, like where the cell is going to go this way or that way, and is that going to be part of the function, or is that separable type of information?
So definitely, I think we need to have an idea of where the cell is going, or if it can go back, or I think it's not just the position, or I mean, I think also going back to your question before, I think it's maybe so it's easy to predict loss of function. I think it's more difficult to predict like a gain of function or like a new cell type, new cell state. So we need to really define not just markers or specific cell type state, but also negative ones. So if we come out with new cells, for example, that we have never seen before, we should be able to see those, identify those, and have an idea of if on the trajectory, if there are cells that should not be there at that specific time is very important. So for neurodevelopmental disorders, we know there are some disorders. One is SYNGAP1, for example. We know that one aspect is that these cells, they maturate much more rapidly. And that's probably the cause of the disease. So we need to know where we are in time, and if we are maybe too late already or too early, or so that's very important.
So I guess what you're stressing is that in addition to the space, we need the time. And that is also a very important element to be able to introduce. And how do we capture things in time in a quantitative manner is also something we need to reflect on with available
time
technology.
And history. So
in history, yes, what happened before.
I think that would be on these aspects, but also and goes in the plasticity of the system. I think there will be cellular states that are absolutely much more plastic, and they will come regularly at different times, and also these more plastic state might be also more sensitive to disease mutation, or to acquire mutation, or to acquire disease states. So I think that will be very important, because might really pinpoint some really critical events over time.
So that's really interesting. So you're thinking that there may be some vulnerability in the developmental system if we can identify these particular cell types that have this propensity to change more easily than others. So getting access to these properties could be quite important.
The way we think is that there might be some cellular states that are actually more sensitive to environment, so that it's just the same environment, they might be more receptive to certain environmental cues, some different morphogen, and so that states at that point might also be more sensitive to stress, or they might be more sensitive, and so they might be able to acquire, they might be acquiring this mutation faster. And we know it, for example, for regeneration is clearly like this. So we know that some regenerative states that are very transient states don't have cell cycle checkpoints. They are very sensitive to all kind of input that is important for the differentiation and to normal sulfate acquisition, but they're also much more sensitive to mutation and to acquire cancer states.
Yeah. And probably organoids and organotypic slices will be key to define the history and the trajectory, because I think it was mentioned it might be hard to extract from in vivo data.
Also because these are very transient and rare states, so there might be few cells, and then in organoid we could be able to enrich for these cell types, so have more measurement to look at the dynamics and of these states.
Yeah. So you've put forward checkpoint, and checkpoint is highly considered in the context of cell cycle. And well, the cell cycle aspect has not been brought up all that much in the discussion. So my question is then, how do we take this into account? Because you have the, what kind of cell you are, but also your properties with respect to cell cycle, entry, exit, and where you are and what you can do. So,
just as that point, I think that a lot of single-cell RNA-seq, the cell cycle is regressed out.
That's also why I'm
then it's very hard to look
sort of jumping into the thing, because I'm just thinking that there's a part of information that we are losing, and that could actually be very important for what we are looking at. And so how can we get back to that and take it into account? Any idea? Yes.
Actually, it's a question on another point. Can I go forward?
Sure. Well, the questions that do not have answers are recorded, so we keep that, but go ahead.
My question is on the adult model that will be built up. And it has been mentioned, the problem of the heterogeneity among the different individuals. And in addition to that, I wonder what about the age of the model, and if it is considered, that's why it's a little bit out perhaps of the scope of this special SCA, but what about aging and, I mean, even in late aging because of the degenerative processes, and might not be so absurd to think about that in this context, because some processes developmental processes seems to be improperly activated during aging. So it might be interesting to make the link, but I don't know in terms of effort how much it will cost and in time.
Anybody wants to comment?
So I'm not sure I understood correctly the question. So you want to have the age of the mother and the context of the fetus?
No. Sorry, I didn't explain. In the global model of the adult, if we consider the age that of the multiple individual that will give that, and I don't know if it taken 30 years, or if we consider also individual of older age for the model of the adult.
Yeah, I think we need actually to consider all kinds of variability. I'm not sure how feasible that is, but for sure we, not just in terms of aging, but in terms of variability of what is normal.
Yes.
So, I think simply having an information about everything, also the age about, is very useful to define that boundary. Not sure if it answers your question.
Yeah. So. Well, but it is important also to have raised the point of this diversity in terms of the dimension to take into account. So I think we are running out of time. I think we have raised a number of questions, and that's probably the purpose of the breakout session. And the future will give the answers, obviously, but we will try to build on that. I want to thank you, all the leaders, for stimulating the whole group, and we have time now for a little break, and then we can reconvene. And so enjoy the break. Thank you very much. Oh, okay. So you want to announce?
So again, you have the list of the 3 breakout sessions for this morning. So clinical genetics here, and then the 2 other sessions in room 106, 108. So you have like a 20-minute coffee break, and then you have to rush to the rooms. Okay? Coffee is ready outside. Yeah.
Start. Thank you very much for all of the session leaders, the breakout session leaders, for frantically putting together the slides for this session in a short amount of time. So I'm Gary Bader, and I'm just going to moderate this session as we did for the breakout reporting back this morning. So we'd like to start with the first breakout session on clinical genetics. I'm wondering if the breakout session leaders can come and present their report back.
So hi. So Mike and myself, we moderated the clinical genetics session, which was quite well attended. The room was not ideal to have many spontaneous interaction, but we had a very good discussion. We discussed 5 key points. Could we have the slides, please? Okay. So we had 5 key points to discuss, and this is on the next slide. Thanks. So first, we discussed the use of single-cell omics data, transcriptomic, epigenomic, and proteomics data in a clinical context, in relation to pathology. And on the next slide, we see what we have discussed. So there are already many single-cell resources, mainly generated in research projects, and we could take advantage of this wealth of data to map disease states. The pitfall in diagnostics, the pitfall in clinical genetics is that we often don't have access to biologically relevant material. The clinically accessible material, such as PBMCs, fibroblasts, muscle tissue, for instance, is not necessarily relevant for the disease. There are examples of diagnostic use. There was an example, a very nice example of spatial transcriptomics of bone tumors, where spatial transcriptomics was really used in the clinical pipeline at the diagnostic level, but also at the prognostic level and the therapy response. There are many advances to be expected in the field of pathology and combining with other tools, such as machine learning tools. And in pathology, we thought that this technology could really be game-changing. In the context of rare diseases, there is a place for single-cell omics, especially in the context of multi-omics, as an additional layer to add to the genomic level, for instance, could advance diagnosis of unsolved cases. Also, there is a big place for the single-cell omics in understanding mechanisms of disease in a low number of patients, and then also to predict disease outcome and to predict the onset of disease and also therapeutic responses, and also to predict windows of therapy. And then for the next questions, I will hand over to Mike.
Yeah, so in the second question, it was more focused on what can already be done, so in the terms of using single-cell multiomics data back to the patient, and what's missing to move forward. And so we've combined this question with the last question, which was the use of machine learning algorithm to be applied to single-cell multiomics. So basically, what's important to discuss is, first of all, there's more and more papers, reports, consortia put together where people are dealing directly to use biological samples, either cell suspension or tissue biopsies directly from patients to try to better understand the physiopathology and find biomarkers. Towards a more use of single-cell multiomics in a routine, we still think we're missing more some standardized pipeline and methods, both wet and dry, so to generate the data and then how to analyze the data in order to be able to, for example, compare the data from different centers, different hospitals, if we want to use single-cell multiomics data directly for diagnostic. And we also discuss about the needs with actions concerted with clinicians, you know, to really from your phone.
Yeah. Ah, okay, okay. Yeah, telephone. Okay. Yeah. Good.
And so basically, what was interesting also so we were asked the question, so are we there yet? Are we doing diagnostic, prognostic, or even like therapeutics using single-cell multiomics? So there are quite some translational research projects going on. There's some published paper, but there's no real established, already existing platform where in a routine a patient's going to come, we're going to generate single-cell multiomics data, and this is going to lead to a diagnostic and so on. So we discussed quite a bit about spatial transcriptomic, which you now generate single-cell multiomics data from a tissue biopsy. So we also think that's going to speed up a lot the use of single-cell multiomics in medicine. And at the end of the day, if we want to speed up the use of single-cell multiomics for back to patients, there's a real need for collaboration between clinician, geneticist, biologist, pathologist, bioinformatician, physicist, and system biology. When we discuss about machine learning, it's a total different field with different skills, different expertise, their own specific questions. So we are not there yet, if you ask us, but there's a lot of translational research projects that are showing us the use and how good it could be to use single-cell multiomics back to the patient.
Okay, the next question is a bit repetition. This is about direct applications of single-cell omics in diagnostics, prognosis, and therapeutics. For rare diseases, we see a place for solving diagnosis, identification of biomarkers, and also stratification of patients, for instance, for therapy, to define the window of opportunity for treatments, for instance, for gene therapy. For pathology, as I already said, there's a big place for spatial transcriptomics, a big opportunity. And then also the PDX models were suggested as interesting models to work with and to encompass the problems of the clinically accessible material. The PDX models could help solving this. And also organoids, of course, good pipelines to develop organoids, IPSs and organoids for patients could also help advance the diagnosis of patients. And then the last question is for Mike.
Yeah, so how to integrate molecular data with already existing diagnostic methods. So we touch a bit on these aspects. Of course, the idea is not to replace, existing clinical and genetic data, but then to build on clinical genetic data and to combine them with data generated at single-cell multiomics levels to try to see what those type of data could bring to us. And there were several examples already of what we could call personalized medicine, by finally looking at single-cell data in the blood or in tissue. We are already able to identify like patients that were diagnosed with a specific disease at the clinical level. And then at the molecular level, there's quite some heterogeneity. So I think this is something important. What single-cell multiomics could bring is to redefine, revisit a little bit the clinical and genetic stratification of patients to focus more on the molecular pathway dysregulated. Because at the end of the day, if you want to get back to patients with a treatment, this is the molecular pathways that you need to treat. And so we all realize that at the clinical level, it could be pretty similar, but at the molecular level, it could be quite some heterogeneity. So I think this is where single-cell multiomics data could bring a lot combined with genetic and clinical data. And I think that's it for us, right?
Perfect summary. Okay.
Thank you very much. So we're going to move to the next breakout session, and then afterwards we will have time for discussion among all the breakouts. So I guess it's Fabien and Paul for the next breakout session on regenerative medicine.
me okay? Okay, got it. All right. Okay, so my name's Paul Riley, and with Fabien Legrand, we led the how the developmental cell atlas and regenerative medicine and the combination thereof, and it was an excellent discussion, although assimilating the 10 pages that followed that discussion was like doing a grant application, last-minute deadline and so on, but it was a lot of fun, and I want to congratulate the participants on the back and forth. We posed a number of questions. We gave some introductions from our own areas. Mine is cardiovascular development and regeneration, and Fabien's is muscle development and regeneration in muscular dystrophy. And we posed the following questions, which I won't outline now because our discussion in the next slide kind of touched on all of these in various aspects. So I'll touch on the discussion points and what emerged from those, and then Fabien will tell you how we feel, or the collective group felt, that the human cell atlas might impact on some of those discussion points and might actually help as we move forward. So it's fairly clear that there is a reactivation of developmental gene pathway in disease that cuts across organ systems. We had a number of experts in different organs in the grouping, which were able to confirm that. In the case of cardiovascular, that is a fetal gene reactivation program, which is really used as biomarkers for many of the structural cardiovascular diseases and post-myocardial infarction heart attack. But they really are just used as biomarkers. So it's an attempt to compensate for the loss of function and the pathological remodeling, but clearly doesn't go far enough. So reactivation of developmental gene program is a signature of disease. It cuts across different organs and disease types, but it's quite clear from emerging studies that whilst key developmental genes are reactivated, they're reactivated differently, and that is borne out of the use of different enhancer regions and co-opting of different transcription factor binding, as an example. So therefore, we do need the integration of the ATAC-seq methylome type epigenetic profiling in order to layer onto this how we understand this reactivation. We had a good discussion about what specific stages, developmental stages, we might need to map onto regeneration, because this could be developmental, it could also be postnatal, and that remains an open question, but of course it's subject to resource and the ability to track that. We need to factor in postnatal development and continued organ growth and maturation. This informs on adult regeneration, so that postnatal period is important, and of course different organs mature differently. There's the usual complexity of multiple cell type interactions, so cell crosstalk, cell signaling, and the niche. And then there was there was a very good point made about looking at single gene expression profiles, differential gene expression pathways, but actually trying to map the gene regulatory networks, and not least because of this different regulation signaling inputs that will happen in development versus disease and regeneration. The spatial and temporal components are obviously very important. Development is relatively long in the scheme of things compared to the sort of firefighting that happens with regeneration. And that's proven by the fact that there's genetic redundancy in development, whereas there's limited redundancy in the regenerative response. So timing of intervention, our understanding of that timing is everything. If we just come to kind of more regeneration-specific components and how they relate to development, the immune response and the inflammatory response is obviously important. The reparative fibrotic response is obviously important. These are things that are not particularly evident in development, of course, but there are cell types, such as the fibroblasts, which make up the skeleton of most organs and signaling components, and of course tissue-resident immune cell types, such as tissue-resident macrophages, which can map across. And the question is, how do they evolve over time to then become contributing to organ formation, to then contributing to disease processes, and can we learn from those developmental cell types as to the sort of phenotypes we might want to modulate in terms of regulating both the immune response and fibrosis, neither of which we want to block completely because that's detrimental to the regenerative response, but at least to modulate. And then there is the differences between organs and the also potential similarities. So in this case, we would need to profile different organs to get a handle on the common pathways for fibrosis and pro-fibrotic signatures, but also some of the important differences could be of interest. And thinking about the extracellular matrix, which of course the efforts to single-cell sequencing do not capture, and the ECM is clearly really, really important, not only in development, where it's absolutely important for signaling components and laying down the structure of where cells can organize themselves and pattern, but also very important in the context of the fibrosis and the rearrangement that occurs in the matrix, that continuous turnover that happens following tissue injury. Cell senescence and aging is obviously important. You can argue aging starts in the womb. Many people do, and there's an interesting component of that. And then, you know, the idea that is regeneration equivalent across organs in the context of the way that tissue assembly and tissue restoration occurs. But the fact that organ maturation is very different as we move through different organ systems. Certain organs become more functional early on and then mature later. Some are functional later at later stages, postnatally and beyond. And then, of course, the insights into individual variation, we heard about that in the context of this morning's breakout, feedback of how individual variation may be a major player here. And, of course, we don't really have a true handle on whether that maps onto regenerative potential and how that then might map itself onto variation in development. So I'll hand over to Fabien.
Thank you. Okay. So that was a really nice discussion, and what I get from it in a nutshell is like we want to do a lot, we want to learn a lot, and as Paul highlighted, we are all focusing on different organs and at different time point during development and on different specific disease. So that's a lot of data sets. So one of the main questions was, are developmental stem cells the same as adult stem cells? So we want to be able to compare atlases of embryonic cell types with growing postnatal, adult regenerating, and atlases of like diseased tissue to try to find if there's commonalities or differences in the stem cells that mediate development, growth, or repair. And to this, we need to compare the expression profile and the epigenomic profile of either developing, maturing, or regenerating tissue stem cells across stages and across ages. And I think there's a need for the community to develop like really robust bioinformatic pipelines so we can integrate many cell types at many different stages and try to find what is common or different and how we can maybe find new tools to restore a pathological organs towards and maybe try to cure it. So, and we discussed a lot also about how can we use the cell atlases that already exist, because a lot of work has been done on many different tissues and many stages. So shall we intersect the developing or the regenerating atlases with the aging cell atlas? That's an open question. And how can we integrate different developmental stage and different tissue all together? We also need to try to characterize rare cell types. So there was discussion about senescence or quiescence, all these intermediate and very rare states of rare stem cells. So how can we see them in an atlases, or how can we highlight them? So there's a need for technology to be able to calculate weight matrices or specific identification of rare cell types that we can study them across age and across tissues. And we think it's a challenge right now to be able to bring together many different data sets to try to identify ways to pinpoint regulatory pathway, being transcription factors or microenvironment or inflammation, that can be targeted maybe to cure disease. But the tools needs to be developed, and I think that's a good goal for the next year, to be able to look at many different cell states and pinpoint it. Is that right? Yeah. Thank you.
Great. That's everything. Okay, thank you very much. So the next report back is about models, organoids, and with a focus on animal models. So I'd like to welcome Matias and Shahragim to present that.
Thank you very much.
Okay, so hello everyone, I'm Matias from Heidelberg, and I moderated the session together with Shahragim from Pasteur. And can I have the first slide, please?
We have to advance it ourselves.
I do it.
Somebody, okay.
Okay, so we put up the following discussion questions. How can model organism help to unravel the human cell atlas? And I think this was the main question we discussed, how are model organisms used to improve the human cell atlas project. A lot of things are done in humans. Nowadays it's possible, but you still need the model organisms, and how are they needed? How can model organisms be used to better understand genetic diseases and also developmental disorders? This is particularly important in human genetics, where you sometimes have to understand the clinical impact of a genetic variant, and you have to decide what model organism to use, or what organoid to use also. And third question, what cannot be modeled in organoids, but in model organisms? We discussed a lot about the limitations yesterday and this morning of organoids, and then the question is now, what are model organisms providing to help this out? And finally, what are the limitations of model organisms? Obviously, there are a lot of them, as they are for the organoids. This was the final question.
Thank you, Matias. Okay, if you go and switch to the next slide. So some of the discussions were on a practical level and scientific level, and some of the discussions were on the philosophical level of strategy and integration. And we started off by giving two tissues. One, I don't want to say simple, because I'll be insulting myself. Skeletal muscle, about 10 cell types. And kidney, perhaps more complex, with over 25 cell types, and there are organs and tissues out there with many more. And one of the challenges, as Matias was just saying, is how to bring that knowledge from model organisms to our understanding of human, because there are going to be similarities and there are going to be diversities. There's some subcellular elements that will be conserved in the fly, for example. We discussed that in the context of the kidney. And that would be difficult, or for the time being unmodelable in an organoid. So one would turn to that model organism. So anybody who's not working on a model organism that's not on the slide, please forgive us, because the slide is limited. Just a few of them were linked and indicated, and as you see in the last column, it was very much a human organoid type of review that has all the tick marks. But clearly the organoids are not recapitulating everything. So one of the key points is, to what extent is morphology and morphogenesis recapitulated in that particular model organism? And one of the examples that come up is that the mouse, in some cases, is fantastic. It has genetics, but the chick and the rabbit, for example, in terms of early embryonic development, more resembles the human than does the mouse. There are orientation problems, there are tissue layer problems, etc. So mouse is heavily used, and clearly in those cases, it's better to turn to the chick and to the rabbit for those early questions. And are the populations similar? Clearly in a fly, on a C. elegans, you have a much less complex tissue. So which of those cell types are conserved? From the stem to differentiated cells of your tissue-specific cell type, and from the context of the niche. So we heard about some of that yesterday: immune cells infiltrating, or stromal cells that are collaborating with the tissue. And if we go down from the cellular level to the molecular level, which mechanisms are conserved? So perhaps that would be easier, because there are a lot of genes that are conserved, key regulatory genes and transcription factors, signaling pathways that are conserved in tissue processes for specific tissues that are conserved across phylogeny. And we heard about this yesterday: timing can be vastly different in non-primate models, timing in a mouse, and an elephant, a human, a chick. And to what extent is that, and the rapidity to which, and the sequence in which those organs appear, important? Okay, next slide. So we identified a few, but not, we didn't list all of them, bottlenecks. One of them is really providing a framework for cell identity across species. And there are going to be gaps from some species, and there are going to be some informative cell types that could be pulled out from Drosophila that would inform on function in human, for example. And then beyond that, how do you integrate that data and knowledge? Because the community is parked in their own niche, and eventually you'd want to bring that in a usable way if we want to inform this human cell atlas. So how do you integrate that data and knowledge from model organisms? Because you would have to appeal to the specialists who are not necessarily accessible or willing, perhaps, or have the time or the resources to integrate their knowledge into the human cell atlas. And something that came up yesterday, benchmarking. Each system would separately need to benchmark with its in vivo state, and that's absolutely critical. And the best teacher, obviously, is the embryo or the in vivo state. And if we want to look at ex vivo systems, we need to keep going back to the in vivo state. So whether we're talking about the human, model organism, or an ex vivo system, each of them have to have some sort of benchmarking across to the in vivo state. And there are issues raised about the epigenome as well. To what extent is that epigenomic signature being conserved or being deregulated in the ex vivo system, or different in a model organism as opposed to human? So clearly some things will fit and some things will not. And one of the discussion points was, is there a phylotypic stage that we can focus on where there is more conservation, before which there's diversity, after which there's diversity among the species? But this famous hourglass design that I took this from Denis Deboul's review from von Baere's notion back in the 1800s. Is there a phylotypic stage that we can focus on if we're doing this cross-species comparison, and then diverge from that, either move forward in development or move backwards into development, where we can have comparative anatomy?
Okay, so yeah, thanks to a good discussion of the audience, we came up with some possible solutions for these bottlenecks. So first of all, we may have to anticipate problems and learn from previous setbacks. For example, we can learn from, we discussed the similarity between the human genome sequencing project and the human cell atlas, and wondered why there was a lot of collaboration in the human genome sequencing project to get to the ultimate goal to sequence the first human being. And this could be maybe applied to the human cell atlas with the help of model organisms. Setback could be also maybe the example of sequencing cancer cell lines. You have this idea, or this example.
Is there an alternation? Okay, somebody's cutting my speech. Yeah, yes. So thanks, Matias. So those two examples. There are many examples out there of mega projects that came up, and there were clearly successes, and clearly there were setbacks or things that were not anticipated. Sequencing all these cancer cell lines in the early days, was that really useful? I personally think no, because they're sequencing derived states until people started to look at cancer-initiating cells. So, and the Human Genome Project, it included certain individuals, it excluded some critical individuals in the initial project. And perhaps we can turn to those and try to anticipate. And a big part of this is trying to anticipate the flood of data that's going to be coming, and make sure that it integrates properly and make good strategic decisions.
So some suggested strategies is to focus on the hourglass model of development, like Shahragim just introduced. So are there common phylotypic stages that we could focus on to understand the similarities between human development and model organism development? And maybe there could be some consortia, this idea came up, for deciding how to integrate cross-species in an organ-specific way. So what are the molecular, cellular differences and similarities? This is maybe something that each field for each organ should decide. For example, in the kidney field, it is maybe interesting to understand what are the important hallmarks of a frog development. We discussed a lot about Xenopus, by the way. And then also compare this with the human kidney development. So maybe this can only be done for people who are working on a specific organ, and then they have to come together and interact. And finally, by doing so, we may understand or come to a holistic view of data from different systems, and also for disease modeling. So there was the example for disease modeling that was a person in the room working on pediatric gliomas. The question was, what model system should be used to study these pediatric gliomas? They first used the organoid system to have a cancer emerging in situ in the organoid. But then they realized they have to go back to the mouse to understand this in a holistic view, to integrate the environment, immune cells, and whatnot, to better understand the disease pathogenesis. So in isolation, these things are very difficult. You cannot focus on just organoids, just model organisms, if you want to obtain informative knowledge, especially for human development. And that's why it's sometimes necessary to interact and collaborate, because within one lab this will be very difficult.
Voila. I think that's it.
Okay, thank you very much. I'd like to now invite all of the breakout session leaders to the front, and we can take questions and further discuss. So it sounds like there was a lot of really interesting ideas put forward. It's interesting to see some similarities across the breakout sessions as well, like for instance the idea that you need to integrate all of the data across not just development in pediatric and adult in the human, but also model systems, and that identifies massive computational challenges that are going to be required to solve to allow people to query all of that data to answer their questions and try to find patterns that are similar or different, and the effects of mutations and need for whole genome sequencing to be integrated, for instance, as well. So right now, I'm just, so thanks for all the summaries. Are there any questions for any of the breakout leaders, any of the topics, any new ideas?
Well, maybe I can start to get people to be warmed up. I was just thinking that there's one aspect that with respect to model organism, we could talk about diversity of organism, because we are restricted here, and I think that the evolutionary aspect has been extremely important in the developmental field to understand how a certain number of processes in particular tissue formation were key. And so I'm just wondering whether that type of thinking could also be inspiring for the human cell atlas in some ways. So I don't have an immediate answer. I'm more raising a question to sort of try and use other ways of looking into things. So I don't know if people have any ideas.
Maybe I can give an example. It comes down to form and function.
Exactly.
And I can pull out the example of the zebrafish, where there's the caudal hematopoietic organ, which is not present in mammals. And you have this migration of hematopoietic stem cells coming out of the dorsal aorta to the caudal hematopoietic organ. And that was definitively shown by live imaging in the zebrafish. And it made a hallmark paper in the field. And there are accompanying papers that corroborated that evidence. So clearly, the form is different, but the function is similar in a way. And on the cellular level, emergence of this cell from an epithelium to mesenchymal cell and migration is a process that was conserved across the amniotes. So yes, I speak to that point. The evolutionary part is important, and there are going to be modules of form and function that could be uncoupled or coupled that would be quite informative.
So maybe that's an area where a bit of thinking could be actually useful to see how we can exploit this sort of approach in a clever way.
Thank you. We noticed that in the session Clinical Genetics, we overlooked the model organisms, and it was very nice to see your table, the overview with pros and the cons of every model organism. And yeah, also in the context of mechanisms of disease, it's a very useful way to move on to go to model organisms and also to discuss this with experts in the field. So this is what you have said. And we also discussed it that we should make a tailor-made approach for every disease, for every question and every organ. It's important to have experts on board.
I think also, if I may comment, what's interesting is like every time you're asking a question, like in biology, it's like if you have a question, you use a technology, you have some data. There's things you understand, there are things you don't understand. Most of the time, what you do, you try to ask the same question using different approaches, right? Mm-hmm. And so I think here is the same with single cell. It's like you can look directly in data from patients, then you can address the same type of question in models, and then you can cross all the types of data to see what's left. If you find directly from patients, like metabolic disease, you find a certain pathway, among others, maybe you're going to prioritize the one that you're also looking in models that you can even see sometimes in development or something like this. So I think at the end of the day, the more we're going to be able to cross different ways of using those single-cell data in patients, in models, in culture cells, the better it's going to be, and we can use everything as filters. Even sometimes you can use data from patients to see how good are the organoids, how good are the models generated of the animal models, right?
And that came up in the regenerative medicine case as well. But maybe in the future, some of these breakout sessions could be reorganized. So maybe there be like a clinical genetics and animal models, for instance, breakout session. I don't know. If people have ideas for that, please post those ideas in the final survey or send messages to the organizers.
I think there are a great deal of excellent specialists working on their specific tissues and organs. And for me personally, the literature is not a place where I can go because I'm just saturated. So a lot of this is lost in the literature of specialists working on their model organisms, and the human people working on the human are not necessarily going to go look at that on that literature, even though they have their keywords coming out. So I think at a higher level, we need to integrate evolutionary developmental biologists in a way that is, let's say, communicative to the Human Cell Atlas, and to be informed strategically, because a lot of that information, which is very rich, is not being exploited.
Great point. Any other questions? I think we start a little bit later, so we can continue for a bit longer.
Oh, where? Sorry, I didn't see. Oh, okay. I come.
Yeah, just another comment to add to the discussion of using model organisms and I guess, in Evo-Devo to inform the Human Cell Atlas. One thing is also to see what's missing in the Human Cell Atlas compared to other animals, and I think that would be really useful in regeneration, like I'm trying to understand comparing the Human Cell Atlas to animals that can regenerate, Paul, your work with the zebrafish, right? Seeing how they can regenerate their hearts and seeing why we can't do it, and then using that as an idea to see what's missing in the Human Cell Atlas and what we could stimulate in therapeutics.
Maybe I could follow up on the regeneration point, because it's obviously a really, really good point. Various people do study the different animal models, I guess. Obviously, the zebrafish is one of them, can intrinsically regenerate its heart and other tissues like fin regeneration. And we look at the early neonatal mouse as a model between the first week of life. But in both cases, we're kind of modeling organ growth and responses as well. So it's kind of a little bit different from what we perceive to be in humans. So on the one hand, they're extremely informative because they give you tissue injury responses, but the postnatal mouse, early neonatal mouse, is a growth postnatal development response. So again, mapping back onto the development atlas. And in the zebrafish, they can grow their organs by hyperplasia. So there's this addition of cells and growth in conditions of high nutrient, good water conditions, low density of animals, et cetera, et cetera. So we are modeling something slightly different, but it all maps onto the growth developmental responses that the atlas can give us. So I think there's still the interplay is even more important.
Yeah. Talking about that, how do you integrate also the work on regeneration that has been done in axolotls, salamanders, amphibians, where there's been quite active work, and so I'm not entirely sure how that is taken on board.
So I guess the area that axolotl and salamander and newt, etc., has really come to the fore. I mean, limb regeneration is an obvious one where you can map some of the same pathways. Has been actually around the immune response and how that is modulated quite heavily to facilitate the regenerative response. So there you have cell types that map onto classical macrophage cell types in mammals, but they're molecularly different and they're behaving differently. Again, they're even triggering release from cell senescence and things like that. So there's some interesting pathways. So, you know, there's a lot to be learned from going back to those model systems, particularly at the single cell level.
Yeah, indeed.
So it's a comment, but I think one of the main consequences of recent developmental biology is to enable these in vitro systems to produce or to reconstitute human development in a dish, like either organoids or 2D systems. And thus that we also give to the field some sort of a translational perspective that was not existing before, in the sense that developmental biology before was, you know, the translational applications were more restricted to fetal malformations and so on. And so this all opened this huge field of cell therapy with the perspective which is really coming of age now with the first clinical trials that are starting to give results. And for this field, it's absolutely critical to have a human development atlas and to understand human development. And I think that's one of the absolutely major justification of this effort.
Great point. So I think we're probably running low on time, so we're going to have to go for lunch. Any other last-minute points? Okay.
Just one thing I forgot to put in the report, and I find was very important at the end of the discussion. We are talking about the need to improve the metadata associated with human samples. Because if we provide single cell or spatial transcriptomics, especially from rare pediatric diseased samples, we need to know a lot more about the metabolic status of the donor, their health status, and their age, etc. And even trying to get whole genome sequencing associated with samples that we profile in single cells. So this is I think, outside of our sphere of interest, because it's more ethical, it's more like communication to the public to try to make people understand that a biological sample is not just a piece of meat or a piece of skin, but it has also to have associated all their history for us to get a lot more of it. So maybe also it's a discussion on like how we can forge some language elements and some discourse towards like humanity, like so they help us better and we help them better. Does that make sense? Okay.
That could also be discussed in the ethics section that's coming up in another breakout. Yeah. Okay, so thank you very much to all the breakout leaders. Great job. So we now have the lunch and the poster session, and the schedule for the rest of the day is up there. I think there's one more. Is there one more announcement to make? Yeah. So everyone can hang out. So we just the HCA wants to make an announcement about publication bundles. So this is something that has been organized for quite a while and requested. So the idea is that by grouping our publications together and approaching a journal or a set of journals, we can combine a whole bunch of publications, submit them all together, and there can be a standard process for pushing those through the pipeline. So we're going to, the HCA is going to run a initial call for a first publication bundle, and there'll be additional ones after that. So the first publication bundle will, if you have papers that are in progress and are ready to submit an abstract by December 31st, you can fill out a form online, and there will be these business cards that are printed out, spread out around the conference area that you can pick up to get your QR code to get access to that form. And then there'll be specific topic areas focused on analytical methods, developmental biology, and developmental cell atlas for more in the context of this conference. And so those will all be organized by the Human Cell Atlas Executive Office.
Just reacting on this and considering the point I think that Shahragim made about session, breakout session on different things that could come together, maybe it could be interesting to think for the other bundle to bring things together so that, because it's easier to have group together things that perhaps would not be looked immediately by people from different area, but that could stimulate them. So maybe it would be a nice way to use it to stimulate people from different area. So just a concept for this open thing.
Okay, thank you. I don't know if anyone wanted to say anything else about publication bundles. No? Okay. So thank you very much to everybody.
I have to pick a card.